Hello, everyone, and we are up to our next episode for Share Trek HIV. I would like to invite Raj here. Hi, Raj.
Hey Maha, it's nice to see you, and it has been a wonderful weekend. So today, what are we planning to discuss, Maha? What do you have in mind to discuss today?
Okay, so I have a question which I see in almost all our viewers asking this question. Why is it so challenging to develop an HIV vaccine?
Yeah.
That
So
is foremost in everyone's mind. So could
yeah,
you let us know why it is so?
yeah, definitely. So it's been very challenging, basically because of two reasons. The first thing is that HIV is genetically very diverse, which means there are various variants and substrains in HIV. So, for example, if a individual has been infected with HIV chronically, and maybe they didn't take medicine in time, or they started ART late. Um, Over a period of time, what happens is that HIV replicates very, very fast. And every time it replicates, it's using our human system for replicating its DNA. And that process is not perfectly compatible. So there's a lot of mutation that happens. And as a result of which the virus, if it has been replicating in somebody's body for a long period of time, it creates a whole lot of variants. And it makes it very difficult to have a standard way of identifying all those variants. In other words, just like we have flu, every year the
Correct.
vaccine that is made for flu is made out of a cocktail of two or three different variants, which the scientists think is the most prevalent variant in the particular season. And that is because flu virus mutates a lot. So they pick up the most prolific of the variants, and then they take the antigen from them and make a cocktail of those antigens and create a vaccine. Similarly, for HIV, um, the variance is a whole lot of variance, and therefore it becomes very difficult to create a single antibody or something to target it, because while you target one variant, there are other variants developing and so on. So it becomes very difficult to do that. And that's the reason why we have not been able to find a, a cure so far.
So this is a question which just came to mind, Raj. So any idea how many variants have been detected so far by the scientists? Any idea
So,
on that?
yeah, a very high level idea is there because it's a very dynamic thing. It keeps changing. Basically, there are two top variants, which is HIV-1 and HIV-2. HIV-2
Okay.
is confined to Western Africa for some reason, and one should be glad that that is the case. And
Mm-hmm.
HIV-2 is not as as virulent as HIV-1, but HIV-1
Okay.
is global. And within HIV-1 and HIV-2, there are substrains
Mm-hmm.
which are numerous. We don't have the
Okay.
count, but they can be classified as HIV-1 and HIV-2. So
Broadly classified into these two.
broadly classified. And as we speak, new variants might be created.
Okay. So what are the current treatments available for HIV, and what is the ultimate uh, goal for HIV vaccines as such?
Yeah, so the current treatment for HIV include antiretroviral drug. So there are things called as reverse transcriptase inhibitors, which inhibit the reverse transcription function. And then there are protease inhibitors. There are different kind of inhibitors. Each of those works on different stages of the HIV life cycle. So in terms of the goals of the vaccine, typically the way we understand a vaccine is that it is preventative, which means you give a vaccine in order to prevent a disease from progressing. But in case of HIV, that definition doesn't seem to fit perfectly, because some vaccines are for prophylaxis, which is to prevent, or it's also called PrEP, which is pre-exposure prophylaxis. And then there are some vaccines which are for curing. So the
Hmm.
ultimate goal of HIV vaccines is to provide broad protection against the virus, ideally by inducing the body to produce broadly neutralizing antibodies that can neutralize a wide range of HIV strains. So we are looking at both ends of the spectrum. One is to prevent new people from getting infected, and the other is to save people who are already infected. And when we talk about saving people who are already infected, we are talking in terms of a few objectives. First objective is to prevent AIDS from happening. AIDS will happen if all CD4 T cells are compromised and knocked out, then the whole immune system falls apart. So we don't want that to happen. That's the first objective. The second objective is to keep the viral load under control so that it's not transmitted to other people. So
Mm.
that's the second objective of the vaccines. And
Okay.
for ART or antiretroviral therapy, it keeps the virus count low. When the virus count is kept lower, the infection into other cells is also controlled. So the dormant reservoirs are not increasing, they remain static. So that's the
Mm-hmm.
second one. The third objective is to find out all the dormant reservoirs and kill them, which is where we are approaching towards sterilizing cure. So ART
Okay.
is more of a functional cure where the virus level is kept lower and infection is prevented and progression to AIDS is prevented. Sterilizing cure is where on top of all of this, you're going and hunting all those dormant
Dormant
HIV
reservoirs
pools,
as well.
killing them off so that you can safely remove ART and expect that there won't be any rebound. So these are the different levels of objectives, and we have different technologies which are available, small molecule, large molecule technologies, and then you have got RNA technologies, and then you have got gene technologies, where you go and
Wow.
make modification in CD4 T cell, give it extra capabilities, which is what the what we call American Gene Technologies AGT 103-T does. And then you've got
Mm.
CRISPR editing technology where you go and literally slice the HIV's important components from the infected cells, which is what EBT 101 does. And then you have the RNA variants where you create an RNA which can go into the body and create an antigen for which the body will then start creating antibodies. So you create an antigen in such a way that it addresses multiple strains of HIV, and therefore the body will start creating broadly neutralizing antibodies, which will adhere to a general class of antigen instead of being a specific class of antigen. So that's where everything is moving towards. That's the objective.
Which brings me to my next question, Raj. Now, what innovations do you think have been made in HIV vaccine development, particularly involving broadly neutralizing antibodies, which we popularly know as broad BNABs?
Yes.
So,
So
what
yeah,
kind of
so
innovations?
BNAB, now when we talk about BNAB, the most popular name is Moderna. So whatever I'm going to
Mhm.
speak now is going to apply to Moderna, and mostly.
Yeah.
One of the significant innovations, I think, is the realization that there is such a thing called as broadly neutralizing antibody. Because before that, we were living in a one-dimension world, I may say, just as an analogy, because that's the easiest illustration I can think of. So in a one-dimension world, you are just seeing the front. In a three-dimension world, you start seeing depth and other things. So earlier, we just knew that there is something called as an antibody, and there is something called as an antigen. A virus has an antigen. Find the antigen and prime the body to prepare antibodies for it, and you're done, right? So that's how vaccines were working. When we encountered HIV, we found that it's not that simple because it
Mm.
keeps on mutating and the antigen keeps on changing. It changes a little here, a little there, but it is now unrecognizable to the original antibody. So the original antibody doesn't work. So
So, we have a
therefore,
hide and seek kind of a situation
yeah,
here, right?
yeah, exactly. So what happened is scientists then started doing research, and they found that there were people who are chronically infected, and in their bodies they found what is known as broadly neutralizing antibodies. So from there, they recognized that we could have a different class of antibodies which don't adhere to just an antigen, but it's capable of adhering to not only the antigen, but also a broad range of variants of that antigen. So that's where the concept of broadly neutralizing antibodies came, which can neutralize various HIV strains by targeting the conserved CD4 binding site on the virus's envelope protein. And this site remains consistent across different HIV variants making it a strategic target for vaccine development. Additionally, researchers at Scripps Institute have developed a nanoparticle vaccine displaying multiple copies of a HIV glycoprotein to introduce broadly neutralizing antibody production. So, in the phase 1 clinical trials, this approach successfully generated broadly neutralizing antibody precursors in participants, indicating a positive immune response without adverse effect. So, that's the extent of progress that has already been made successfully. Yeah, so
in the
that's what I
vaccine
would
department.
say. Yeah.
Now, how are researchers transitioning from protein-based HIV vaccines to mRNA-based vaccines? And what are the advantages of mRNA vaccines,
Yeah.
if I may ask? Yeah.
Yeah. So what happens with protein-based vaccines is we are in the one-dimension world where you have a simple, it looks like simple to us now, but when it was first encountered, it was very complicated. Take the case of polio, single
Hmm.
antigen, and for example, and it's working and it's not mutated and everything is going fine. So you just take the antigen and the vaccine is made out of the antigen. So the body creates antibodies for that antigen and the patient gets a lot of protection and the antibody keeps on working, the B cells keep on working as long as they are exposed to that antigen. So if they are not exposed to the antigen for a certain period of time, then what the body does is the body recycles those CD4 T cells, those B cells and all that stuff because it wants to create space for new antigens that the body might encounter. And that's where you have this concept of booster vaccines, where
Hmm.
scientists find out how long the immunity will last in the body if the body did not encounter the antigen or the pathogen. And
Hmm.
at that point of time, they say, come for a booster vaccine. Tetanus, for example, you have booster vaccine after 2 years. So
Hmm.
those kind of things happen. Now, for this, the scientists have to create the protein and then send that protein into the body, and they have to create a bigger dose. With mRNA, what happens is that mRNA conditions the body to produce the antigen within the body using the body's own resources. So the manufacturing is outsourced to the body itself instead of having to put up a factory to create the protein, do quality control on the protein, do purity check and other things, and then package it and then send it as injections. So with mRNA, a smaller dose can achieve the same effect as a big dose of protein-based vaccine. So that's the second advantage. And people will ask, what is an mRNA? So
Hmm.
mRNA is basically the recipe for a protein. What happens is that our human body has got DNA in each cell, and when the cells experience any kind of stress, for each kind of stress, the cell knows which gene has to be alerted. So the cell tells the nucleus that gene number so and so has to be activated. So when that message goes across in the nucleus, the copy of that gene is thrown out of the nucleus, and the copy of that gene is called as mRNA. And mRNA is basically a recipe for creating a protein. And there are components in the cell environment called as transfer RNA and other RNAs. They all work collaboratively. tRNA can decipher the mRNA recipe and asks the cells to provide amino acids, and it arranges them in a sequence so that the protein recipe is completed and a new protein is formed exactly as per the recipe. So that's how mRNA vaccines work. They send the body a recipe for creating a protein or an antigen.
So, Raj, can you share the latest updates from recent clinical trials of mRNA-based HIV vaccines, particularly any observations or challenges? And
Yeah,
you have explained the whole thing very beautifully.
yeah,
You know, people who are watching, I'm sure, can visualize actually what's happening within the body.
right.
Very beautifully explained.
Thank you. I hope people can understand. One of the common problems for the channel basically is that the English speaking audience are able to understand, and the Hindi audience is always saying, Sir, Hindi, Sir, Hindi. So these are all complicated concepts. They involve complicated syntaxes and terminologies. It's very difficult to do all this thing in Hindi. So that's why we have the Zoom meeting, which
Yeah,
happens once every month, where we talk about all these things in Hindi, where I don't have to write a script, but I can talk about it in Hindi. Whereas making a video means writing a script and explaining the whole thing in Hindi. It's time consuming and it's totally
I know,
unviable.
I know. But
So
I think Raj, what you could do is, because you have this wonderful ability to kind of speak in a way that people can visualize, in a very simple way, the way that you spoke about the polio vaccine and
yeah, yeah,
how it is easier there, and
yeah.
why antibodies and antigen. So the way that you taught that concept, perhaps in a small way, perhaps that could be done in the Hindi version. Is
Yeah, and
my 2 cents.
yeah, definitely,
We could try
definitely.
that.
We could try that, and that would help. But what I have done in the past is I have tried to do exactly that, and it has taken a whole lot of time. So
Oh, I know.
for me, I'm going to do a little bit of deviation out here, and I hope the
Hmm.
audience excuses me for this. But for
Yes.
me, what happens is that I have two channels going on. I'm an investor. I'm a
Correct.
retiree. And if I don't keep
Yes.
focus on my investment, right?
Right.
And I'm also a real estate agent. I'm a broker here. So if I don't keep my focus on those things,
I
my
know.
bread and butter gets affected.
Correct. Correct.
Who wants to
Correct.
be poor in old age? Who wants to keep on working when they are 80 years old, right? So I have made, by God's grace, I have made enough money, but my responsibility now is to preserve that money because share
Correct.
market will fall anytime and share market can go up. So my retirement funds are all in the share market and in properties. So I have to focus on that. That I cannot sacrifice. That's my first responsibility is to my family and to
I
myself.
like that line. Who wants to be poor?
Exactly. And my some part of my Hindi audience does not understand that, and they think
Yeah.
that just say Hindi, one line comment Hindi and leave it, right?
Yeah.
And I'm trying to beat it into their head that please understand, be
Yeah.
responsible, right? They don't
And as you
understand.
rightly said, Raj, these terminologies cannot be
It's
kind
very
of
difficult.
translated into Hindi.
Exactly. These are all
There are
difficult.
no
And and
synonyms for it.
yeah, and Maha,
Hmm.
science and technology is not developing in Hindi. Science and
Exactly.
technology is developing in English. So English creates new
Exactly.
words to describe new
True.
context. What is IQ? IQ is the ability to visualize and understand various concepts. And each of those concepts have to be described. That's where language comes in. So a language which is alive and robust is the one which has science, technology, enhancements and advancements happening. And new words keep on popping up. You have to understand the new words. To understand the new words, it's described with existing words. And those are at the higher level of understanding. So for me to drag the Hindi audience and give them all of these things in Hindi is to drag
I
myself
know.
up in my understanding of Hindi and become a Hindi scholar and start using Hindi words to cobble them together. And for example, the Hindi word for railway engine, even now,
Oh,
is a
huh.
Loha Dandamarg something something. It's a huge
Oh
word,
my gosh.
right?
Oh
Where
my. Let's
so
not
I
even
can't
get
do
there.
that, and my Hindi audience is refusing to understand that they don't want to become a member and come into the membership section. And why do I have to do it in member? Because people are coming on video and they're showing their face, their names are there, and they're asking questions, and I'm answering them. It should not be floating around in internet for the whole world to see. Their
Exactly.
colleague or their relative or somebody may see them and they don't know they're HIV positive. How do
Right.
you want to release that, right?
Yes,
So
yes,
to
absolutely.
cut a long story short,
True.
Hindi
True.
audience needs to become become members, attend in the Zoom. I can't explain all this complexity to them. Now, coming back to the
Yes,
question that
let's
you
come
asked.
back
Okay,
now. Yes.
so let us talk about the clinical trials. Okay, on March 1st, a news article in Science detailed skin events that were observed in the HIV vaccine trial. So the IAVI G002 trial is what I'm referring to. You can
Mhm.
always Google that and you can see information if you're English speaking person, even for Hindi speaking person. You can Google that and ask Google to translate it into your language so you can see all the things. And I'm also putting Hindi subtitles in all my videos So you can always use subtitles. Okay, so the IAVI G002 trial was designed to test if sequential administration of priming and boosting HIV immunogens via mRNA can activate naive B cells and guide their maturation towards providing broadly neutralizing antibodies. Now, that particular test reported skin events such as hives. Hives means people break out into red dots on their
Oh,
skin,
yeah, I know that.
itchiness. So that happened in 7% to 18% of volunteers across 3 trials. And these events were generally mild or moderate and managed with allergy medication. And there were no serious adverse events. And this happens with many established vaccines as well for certain number of peoples. And affected participants received necessary medical care and close monitoring. Investigations into these skin events are ongoing to understand their causes and to see if some tweaks can be made to prevent that. But it's not very serious. Yeah.
Okay. Yeah, hives is something that is commonly seen, right?
Yes.
I mean, even for some people, even if they scratch some part of their skin, the skin kind of thickens. Is that happens?
Yeah. So hives happen when you get an injection or you get allergy to something, you see it going.
True.
If you go to poison ivy, or in Hindi
Hmm.
they say khujli ka jhad, it has got leaves.
Hmm.
When the leaves touch you, you get rash.
Correct.
Yeah. So
Correct.
breaking into hives. Yeah. So that's it.
So Raj, can you share some latest updates from the clinical trials? I mean, okay, one was this hives and itchiness. Other than that, no serious side effects were seen, right?
No, nothing else. Yeah.
So, what has been encouraging about the results of these clinical trials now, despite the skin events that we spoke about?
Yeah. So in my opinion, the skin events are all minor because skin events always happen when you're injecting something into the body. There is a good chance it can happen. There is an established anti-allergy drug uh, body of drug. The science is very well established, and we got various agencies for reducing all these kind of skin events and allergies. So I don't see that as a big problem. So I would say that despite the skin events, the IAVI G002 and related trials have shown favorable immunogenicity results, indicating a positive immune response. And the preliminary data on the immune responses and safety have been presented at scientific conferences, highlighting transparency and progress. The focus remains on refining the immunization strategy to elicit BNAbs, or broadly neutralizing antibodies, while understanding and mitigating the observed safety signals. These trial results will be published in forthcoming papers in greater detail, and we'll have much better understanding of what exactly was observed in terms of generating the BNAbs, because that's where I'm more interested. The
Hmm.
hives things and all, they will go away. So yeah, that's what I'm looking forward to, more data release so that I can bring to our audience exactly how many BNABs were generated and how those BNABs persisted, and whether HIV was able to mutate and
Hmm.
outfox the BNABs. So that's what I'm
Okay,
looking
that's
for.
something we need to watch out for.
Yes.
Very interesting. So, Raj, in conclusion, now, what does the future hold for mRNA-based HIV vaccines? And what steps are being taken to address these challenges?
Yeah, so that's yeah. In terms of that question, I would say that the future of mRNA-based HIV vaccines is promising, with significant advancements offering new hope. And while challenges such as the virus's genetic diversity, its ability to mutate very fast, and its targeting of the immune system remain, the virus has not fundamentally changed, even though it mutates. Innovative strategies using broadly neutralizing antibodies and mRNA technologies provide promising avenues. And the other aspect is that if you look at the protein-based vaccines, uh, they uh, they take a longer time to manufacture, and they need a bigger dose, etc. mRNA, the entire life cycle of mRNA clinical test is much faster. So, yeah, and also FDA is getting much more comfortable with mRNA vaccines thanks to the success with COVID. So I think mRNA is the way to go in future, and it provides a promising avenue. It outsources the entire antigen creation to the human body. So it's a much better approach. Ongoing research and clinical trials are crucial to developing a vaccine that can provide broad and effective protection against HIV. And the latest updates from clinical trials emphasize the importance of addressing safety concerns and refining vaccine strategies to achieve this goal. And now I would like to also add into the mix artificial intelligence. In AI, AI is all about matching patterns and learning from patterns. So AI is going to help a lot in identifying all the possible variants of the common proteins or antigens that are present in the HIV highly conserved areas. And scientists will be able to use AI to create antigens which kind of represent a broad range of variants, so that that broad range of variants can be represented by a single computed antigen, and that antigen can be coded as an mRNA and injected into the human body, and then clinical trials can be done to see how the body responds and how the B cells provide broadly neutralizing antibodies in order to provide human immune systems the ability to combat that range of HIV. And then the question will become, if HIV continues to mutate and we cover a broad range of broadly neutralizing antibodies. I'm not talking just about one BNAB, but a cocktail of BNABs, right? And then with every subsequent mutation that it does, and there'll be another BNAB added. So we are in a higher level of playing. It's like a higher level of three-dimensional chess kind of a thing, where our drugs will become even more and more potent. And in the battle, ultimately, HIV has to give up because we have covered all of its. If you're playing tic-tac-toe,
Variants.
right?
Right,
So you
right.
keep on, the opponent keeps on, and finally the opponent is fatigued and you win. It's like
Oh,
that.
that's a beautiful analogy. I mean,
Yeah, yeah,
tic-tac-toe.
yeah.
Yes,
So that's where we are
something
now.
new. Come on, now tell me what are you going to do? Okay, Raj, this was very wonderfully explained, especially the analogy about the tic-tac-toe and the new variants coming in and new BNABs coming to attack it. Let's hope that the clinical trials for mRNA come out successful and without any, let's keep our fingers crossed, no side effects.
Yeah,
And that would be a great thing, right?
yeah. I'm
Great
hoping
advance
for that. I'm
science.
keeping my fingers crossed for that, and yeah, I think our audience should be happy with all the information that we have given so far, while we wait to get more information about the mRNA vaccines.
True.
So I think that kind of brings us to an end of today's session, Maha. And thank you very much for going through all the information that I had sent you and asking me all these questions to bring out the information for our audience. It's really been a pleasure answering your questions and recording this video. Thanks a lot.
Well, if I may take a minute more, I must
Yeah.
say, Raj, that prior to coming to this channel and coming on these various episodes with you, I did not know what an antibody was. I didn't know what an antigen was.
Yeah.
I didn't know what BNABs were.
Yeah.
I didn't know that vaccines are being developed for this. Thanks to you, I'm learning so much. And
Yeah.
I'm sure even our viewers are getting a whole lot of information. And
Absolutely.
actually, I would say that it's not something just hearsay. It is accurate information, which is
Yeah,
they're really
yeah.
lucky to get
We
this kind of
strive to
information.
give accurate information, and also I am not a scientist or a doctor. And then the question becomes, why are you making these videos? It's because the human connection. When we started having hundreds and thousands of people demanding that they want to know more about this vaccine and the disease, I couldn't but help put all the efforts to start learning block by block by reading up on scientific literature and this and that, so that I could develop this understanding. And again, I have not gone into the class to become a doctor or a scientist, so there is a lot of gaps. My knowledge is like it's broad, but it's not deep, and in the breadth there are a lot of holes out there. And I discover a hole when a new therapy comes in using a particular method, and I read about that, then I understand, oh, there was this big hole of knowledge that I did not know. It was a gap of knowledge that I did not know. So then I start reading upon that. So for creating some videos, it's very easy because it's based on my existing knowledge base that I accumulated. And in some videos, it's a whole lot of new learning because it's a new technology. Like when we spoke about HLP, I had to do a lot of reading. so that I could have the confidence to talk to Dr. Eric Arts and ask him
Of
questions
course.
and make sense of his questions and ask follow-up questions. So because those people have very less time in their hand, and in that given 30 minutes or whatever we get with them, we have to ask the right questions, we have to understand their answers and ask follow-up questions. So I'm also learning. We try our best, and rest assured, if we have given you any wrong information by mistake, we will correct it. We'll accept our mistake, we'll correct it. But to our best of our knowledge, we are giving you most accurate information possible. Okay, so that's about it, Maha. Then I think we'll end this video today here now. And big
Thank you,
thanks to
Raj.
all our viewers. Okay, guys, bye for now. We'll catch up with you in the next video. Please don't forget to press the subscribe button and the like button. And I have a request for you. Please forward our channel to all your friends who you think deserve to see this channel. We want to touch the 20,000 target. We are very close to 15 right now. Please take us to 15 at least, and then we'll see what happens from there. Thanks and have a great day. Bye for now.